I am Matt Pillar, Chief Editor at Bioprocess Online, host of the Business of Biotech podcast, and moderator of the Bioprocess Online live series of discussions you're joining today. And on this particular morning, I'm not moderating per se, so much as I am going one-to-one with Astellas Gene Therapies Director of Technical Development, Joshua Zibit. Before we meet Josh, I want to take a second to thank our audience for joining us and remind you that this program is not just for you, it involves you. I'm going to ask Josh some questions, but your questions are far more important to both of us, so please use that chat screen to enter your questions throughout. I'm not going to batch them for the end. I'll address them as we go, and I'll only address you by your first name to protect your anonymity. I'd like to take another second to thank Culture Biosciences as well for its gracious support of today's program. Be sure to check out their tech-centric, cloud-connected suite of bioprocess development solutions by engaging some of the on-screen content offerings that you see. Now back to Josh Zibit. He kicked his work off with Astellas as Director of CMC Operations back in 2018, and he rose through the ranks from there, taking on the role of Senior Director of Global Supply Chain in 2019, and then taking on the role of Executive Director of Gene Therapies, Global Supply Chain, and CMC Operations in 2021, just stacking the responsibilities. He's been in his current role as Director of Technical Development since 2022. Josh spent the bulk of his career prior to that in manufacturing technical leadership positions in Astellas' ADC and antibody efforts, giving him a well-rounded view of multimodality process development and the technologies that support it. That's what makes him a great guest to go mano a mano with. I guess that sounds kind of confrontational, does it? Mano a mano. We're not gonna fight. A great guest to go one-to-one with, and that's why I'm super excited to introduce him to you now. Josh, welcome, and thank you for joining us.
No, thanks, Matt. Very happy to be here, and I think it'll be a fun discussion, and looking forward to seeing some of the questions from the audience today.
Awesome. Good deal. Well, I want to jump right into it. And, one of the promises I think of the program is to talk about some of the advances, tech advances. And you, as I just ascertained, even though you have got a very youthful look about you, you've been in the space for long enough to probably see some of those advances. So we'll start there at a high level. What analytical technologies, tools, instruments, and processes are available to you today in your role that maybe weren't just a few years ago?
No, yeah, thanks, Matt. Over the years, we've seen some significant improvements in the technologies and tools that are available to us. Some of this is just incremental progress that you see where vendors come out with updated versions, every couple of years. But in other areas, the improvements have been more substantial. Obviously, I think everyone's aware of all the news around AI these days. And so I think that's actually a pretty exciting space to monitor and see how that can be applied to bioprocess development. It's pretty exciting. We look, in that area at some mechanistic modeling, seeing whether we could actually create digital twins, modeling based off of the large sets of data that we develop. I think it's still really early, but the promise is there. And, it's pretty exciting because if it's successful, you can both speed development as well as, reduce the number of actual experiments that you need to run in the lab because you can use In-Silico modeling.
Yeah.
So that's pretty exciting. Other areas that keep us interested, we have improved monitoring of cell health for, upstream bioprocessing, including, more extensive use of Omics. For gene therapies in particular, there's improvements in sequencing technologies, so improved long read sequencing, so you can really understand better what it is you're packaging within a gene therapy, as well as some improved digital PCR technologies that simplify the workflow for improved lab operations. Looking at the process development area, there we really try and look at improvements in terms of high throughput screening. And so, yeah, there's newer tools that allow us to do, large sets of experiments, both upstream and downstream, in a more automated fashion, and that really allows us to run, you know, rapid, large sets of experiments, generate a lot of data, and then be able to analyze that to make process development go faster than if you needed to do it one experiment at a time. Some other areas that aren't particularly new, but we're seeing maybe more application is at-line HPLC systems to monitor the process, as well as use of Raman and near IR spectroscopy.